everyone, my name is Naiwich Duop. My group members are Shar Speth, Natasha Lee, and Alejandra Hernandez. Our group will be presenting hepatitis B. Well, I hope you enjoyed the video and learned something new. I'll turn the time now over to Shar.
Hi, my name is Shar Speth and I will be talking to you about etiology.
Hepatitis B is a virus classified as hepatinoviride orthohepatinovirus. The morphology of this virus is enveloped, meaning it contains an outer membranous layer that surrounds the nucleocapsid of the actual virus. Hepatitis B's viral genome is a partially double-stranded and partially single-stranded DNA virus. There have been a few genetic factors linked with the patient's susceptibility to hepatitis B and the severity to which the disease impacts them. These links could work to distinguish why some patients are affected acutely and why others are affected chronically. Specifically, we will talk about TNF-S59 gene, IFNGR1 gene, and IFNGR2 gene. TNFS59 is a gene that encodes the CD137L protein. Patients with chronic hepatitis B infections were shown to have an increase in the expression of this gene. This correlation indicates that patients with overactive TNFS59 gene, if infected by the HBV virus, were more likely to experience symptoms of hepatitis B chronically. INFGR1 and INFGR2 are both genes that contribute to immune system response. The dysfunction of these specific genes has been correlated to a patient's susceptibility to hepatitis B. IFNGR1 is a gene involved with cell-to-cell communication. This gene is commonly activated in response to an infection. When this gene malfunctions, it leaves the host more susceptible to infection. The IFNGR2 gene is similar to IFNGR1 in the sense that it is based on immune system response. Dysfunction of one or both of these genes can lead to a patient having an increase in susceptibility to hepatitis B infection.
Hi, my name is Natasha Lee, and I will be talking to you about the reservoir and signs and symptoms of hepatitis B. A reservoir is a host of a pathogen of an infectious disease. With hepatitis B, the main reservoir is humans. It is rare for any other species to have hepatitis B except for chimpanzees. There are two types of hepatitis B, acute and chronic. A hepatitis B infection is considered to be acute during the first 6 months after being exposed. This is the average period of time it takes to recover from a hepatitis B infection. If you still test positive for hepatitis B after 6 months, you are considered to have chronic hepatitis B infection. This can last a lifetime. Approximately 10% of adults and 25% of children with acute hepatitis B will continue to test positive for the disease and develop chronic hepatitis B. 90% of infants who are infected at birth by their mothers will also develop chronic hepatitis B. Next is signs and symptoms. Some people may confuse signs and symptoms as the same thing, but there is a difference. Signs are visible where others can see there is something wrong, and symptoms are what only can be described by the person feeling them. One sign of hepatitis B is jaundice. This is when the skin is yellow as well as the whites around the eyes. Others are high fever, dark urine, and vomiting. The symptoms can be described with abdominal and joint pain, loss of appetite, nausea, weakness, and fatigue.
Hello, I am Alexander Hernandez and I'll be talking about transmission and treatment.
Transmission. Hepatitis B is a viral infection that attacks the liver and can cause both acute and chronic disease. The virus is transmitted through contact with the blood or other bodily fluids of an infected person. The hepatitis B virus can survive outside the body for at least 7 days. During this time, the virus can still cause infection if it enters the body of a person who is not protected by the vaccine. The incubation period of hepatitis B is 75 days on average, but can vary from 30 to 180 days. The virus may be detected within 30 to 60 days after infection and can persist and develop into chronic hepatitis B. Common ways that can be transmitted. Hepatitis B is also spread by percutaneous or mucosal exposure to infected blood or and various body fluids, as well as through vaginal and seminal fluids, the reuse of needles and syringes. Infection can occur during medical, surgical, and dental procedures, through tattooing, or through the use of razors and similar objects that are contaminated with infected blood. Hepatitis B and pregnancy. If you're pregnant, you might pass the virus to your baby at birth. It is less likely to happen during your pregnancy. All infants receive the hepatitis B vaccine as soon as possible after birth, preferably within 24 hours. The birth dose should be followed by 2 or 3 doses to complete the primary series. If your baby gets the virus and isn't treated, he could have long-term liver problems. All newborns with infected mothers should get hepatitis immune globulin and vaccine for hepatitis. Treatment. A vaccine against hepatitis B has been available since 1982. This vaccine is 95% effective in preventing infection and the development of chronic disease and liver cancer due to hepatitis B. The best way to prevent hepatitis B is by getting the hepatitis B vaccine. The hepatitis B vaccine is safe and effective and is usually given as 3 to 4 shots over a 6-month period. In most people, however, the treatment does not cure hepatitis B infection, but only suppresses the replication of the virus. Therefore, most people who start hepatitis B treatment must continue it for life. People with chronic and active hepatitis B infection can be treated with oral antiviral pills or injected medication known as pegylated interferon.
Hello again, here's some additional information about hepatitis B. Hepatitis B is a common disease in the United States. An estimated 800,000 to 1.4 million people in the United States have chronic hepatitis B infection. Every year, HBV kills approximately 2,000 to 4,000 people in the United States. Their death is usually a result of complications of liver disease. An individual's susceptibility to chronic HBV infection is closely correlated to the age in which they are infected. Only 5% to 10% of adults and children who are infected end up with chronic HBV. 25% to 50% of patients infected under the age of 5 will end up with chronic HBV. As much as 90% of infants infected with HBV at birth end up with chronic hepatitis B. Looking at the geographical distribution of hepatitis B, the sub-Saharan Africa and East Asia have the highest prevalence of hepatitis B infection in the adult population, ranging from 5 to 10% of the total population. The Middle East and India subcontinent have an estimated 2 to 5% of their population infected with HBV. In countries of Western Europe and North America, less than 1% of the population are chronically infected with hepatitis B. Diagnosis of hepatitis B is only possible through laboratory diagnosis. Acute HBV infections is identified by the presence of HB surface antigens and the immunoglobulin M, the antibody to HB core antigen. In early infections, the HBe antigen is usually found. High levels of this marker also indicate high levels of replication in the HBV virus. This indicates that the bodily fluids of this host are highly contagious. In patients with chronic hepatitis B, HB surface antigens is present in the patient for at least 6 months. The persistence of HB surface antigens for an extended period of time leaves the patient at risk for developing chronic liver disease and liver cancer later in life. Some individuals are at higher risk of obtaining hepatitis B infection than others. People who frequently require blood or blood products such as dialysis and recipients of solid organ transplants have an increased chance to exposure. People incarcerated into prisons are exposed to diseases from other inmates around them. Individuals who inject drugs may use dirty needles or share needles that are contaminated with HPV virus. Individuals with multiple sex partners can be exposed to all kinds of diseases unknown to them. Even healthcare workers can be exposed to the blood of an infected individual. Travelers to epidemic areas have an increase of exposure to HPV virus. A hepatitis B virus vaccine demonstration project was conducted in southwest Alaska in 1981 to 1982 to determine the immunogenicity and efficacy of the vaccine. A total of 1,630 susceptible persons in the Alaskan Native population were vaccinated with the recommended 3-dose regimen of plasma-derived hepatitis B vaccine. 94% demonstrated antibody to hepatitis B surface antigen at levels of less than 10 milli-international units per milliliter. After 10 years of follow-up, 76% of those immunized had antibody surface antigen levels at less than 10 milli-international units. During the 10 years following the first dose of vaccine, 13 study participants developed antibody to hepatitis B core antigen, 10 vaccine responders and 3 non-responders, and none developed sustained hepatitis B surface antigens positivity or had clinical hepatitis. These data suggest that immunization with hepatitis B vaccine continues to provide high levels of protection from clinical disease for at least 10 years. This concludes our presentation for hepatitis B. This is a slide of our references. Thank you for watching.